Welcome to Data in Biotech, a podcast from Cordine where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. Today, we're joined by Patrick Torbey, CEO and co-founder of Neoplants, a synthetic biology startup based in Paris. Patrick and his team are pioneering a bold new vision, engineering indoor plants and their microbiomes to purify the air we breathe. In this conversation, we dive into the science behind volatile organic compound degradation, the challenges of genetically modifying ornamental plants, and the role of simulations and testing in scaling bioengineered products. Patrick also shares how Neoplants balances deep tech R&D with go-to-market strategy and why he believes that synthetic biology could hold the key to solving global environmental challenges. If you're interested in the future of plant design, indoor air quality, or applying data science to biotech R&D, you won't want to miss this episode. Here we go.
Patrick Torbey, welcome to the Data in Biotech podcast.
Thank you very much.
Well, awesome. Just to kick us off, would you mind giving us an introduction to your background and what brought you here today?
Yeah, sure. So I'm the co-founder and CEO of a biotech company called Neoplants, and we're specialized in the engineering of plants through their genetics and their microbiome to have a positive impact in people's lives.
Awesome. Why don't you just introduce us? What are you genetically engineering plants to do currently? And, what are the plants that you have on the market right now?
So the main problem we're tackling is the problem of indoor air pollution. So the air that we're currently breathing, you and I, indoors, is 5 times more polluted than the one we can breathe outside on the streets, even if you live in a big city. And that's due to a specific type of pollutants called volatile organic compounds, things like formaldehyde, benzene, toluene, xylene, nasty compounds emitted by everything you have indoors that makes the air quality really bad. And the problem is that there's no good mechanical solution for that. Normal air purifiers filter out particulate matter, but can't really handle VOCs in a real sense. And so when we talk about volatile organic compound, organic means carbon, and plants love carbon. So what if plants could absorb these toxic chemicals and metabolize them? And how can we create a product out of this technology? It's not just about the science, but how do we democratize this to people, to put it in people's homes? So this is kind of what we're tackling.
What are some of the biggest scientific or engineering constraints that you had to face when you were thinking through the problem of how to engineer a plant to improve indoor air quality?
The main difference between academic research and a startup is that you are creating a product. We're not just trying to have results. So you need to start from a plant that people already buy. And so you start with a plant that science hasn't researched before, because nobody researches the physiology of indoor plants in a deep way. So we started with the selection of the plants, and we decided to work on pothos, which is a monocoque vine kind of plant that is one of the most, if not the most sold houseplants in the US. But to be able to genetically modify it, first you need to sequence the entire genome, which hasn't been done before. So we did a de novo whole genome sequencing of pothos. I think it was the first whole genome sequencing, at least with publicly available information of indoor plants ever. So that was very fun. And then we start to study it. So what's the transcriptome? What's the proteome? What's the metabolome? How does it actually work? And what type of enzyme do you need to introduce to bridge the gap if you want to go from formaldehyde to sugar, or from benzene to an amino acid? And from there, you say, okay, what are the available enzymes you have within nature? There's the second challenge here, which is most of these enzymes exist in the prokaryotes, so bacteria. So you need to translate these genes and make sure they work on the higher plant, make sure they're expressed at the right level, make sure that they actually don't have a byproduct when they actually do things efficiently. We also worked in parallel on the absorption of these pollutants. What if the plants had more stomata, more pores that allows for gaseous exchanges, so it could absorb these VOCs a bit faster? So these were the two pathways we saw how to engineer the plants. And we're 5 years down this process. It took a lot of time to understand and then engineer it. But now the plants are now growing in the lab, which is great. Same thing with the feature, like the aesthetic feature of the plant. We haven't announced it yet, but we also had to look within nature, like how can we change either the the color or the shape or the fragrance, or what could we change on that plant and how to make it radically different. So that was also something that's not explored by the literature. You find kind of hints here and there, experiments that weren't supposed to do something, that did something interesting with the physiology of the plant, and just explore. So there's a lot of creativity involved, a lot of luck, because this is empirical science, because there's very little data to go on. So it was a big chunk of our time gathering this data, this knowledge, understanding the plant, and then engineer it. And so this is how we went through. Like, these were the challenges, or I would call them the opportunities, like the pathways that are open for us to create this type of plants. And the same pathways are also open, and actually, the more we delve into it, the more we thought they were even more interesting, which is the microbiome. It's interesting. We thought that the microbiome would have a lesser impact than the genetic engineering of the plant itself. And what we saw is that it could be so powerful, so much more powerful than what we thought, just the microbiome alone. So that was also a journey of discovery, because nobody tried to do this stuff before.
Yeah, that's awesome, and I would love what I'd love to do is sort of dig into the specific degradation pathways that you're targeting with the plant and the microbiome, and then talk a little bit about your optimization process, how you gathered data and how you got down the path of understanding better what was going on. So my understanding is that you've got two degradation pathways. You've got formaldehyde, and then you've got BTEX. And then, your goal is to basically take as much of that out of the air as possible, to just like optimize. I think you refer to it as flux optimization, of, just getting as much of that out of the air and into the plant itself, or into the plant microbiome as possible. I mean, I think what I'm interested in is, how did you come to the conclusions about how you were going to do the degradation? Were you looking at the enzymes that were out there and using those to sort of guide what was possible to connect these pieces together in order to end up in something that was consumable by the plant or the plant microbiome? Or did you have in mind, that this was the path that had to be taken from a chemical perspective, and then go out looking for the enzymes that were going to sort of enable, or the processes inside of the plant or the microbiome that were going to enable that to happen? Or was it some combination of the two?
We started with, we need to go from benzene, let's say, to something that the plant can degrade. So we looked at all the available data sets online, like enzymology data sets. What are the enzymes that can turn benzene into anything? So you get to this first circle. And then, do we have anything that the plant can use there? Not really. So we have to go to, okay, from all these molecules that can be produced by benzene to something else by anything in nature, whatever the organism. Okay, first, like with one enzyme, we can't get there. But with two enzymes, we have a few different candidates. And catechol for us was the most interesting. So you get from, okay, we could go from benzene to catechol, and it's a two enzyme step. But there's a lot of different enzymes from very different organisms that can get you to phenol and get you to catechol. Some actually is one single enzyme that can do the two steps, actually. So you test those. You say, okay, let's have all these different types of enzymes from a lot of different organisms. And the more the protein sequence differs, the better, because you can have a diversity of results. Test all of those inside plants, and then see what works, what doesn't, through analytical chemistry. So you can trace where does the benzene go, and how is there a bottleneck at step 1 and step 2. And then you get to making sure that this is as smooth and the flow is as easy as possible. So you take, okay, these are the best enzymes. Is there a bottleneck there? Do I have to do protein engineering? And in our cases, we also did that. We also found different ways, like different enzymes where they didn't even take benzene, but they took something that looks a lot like benzene, and we did protein engineering to see, okay, now, because either you start with like basically bacterial enzyme that do exactly what you want to do, or you start with a plant enzyme that doesn't take benzene as a substrate, but takes something that's very, very similar. And this is still confidential information, so I can't go into the detail. But you change the specificity of that enzyme to allow it to take benzene as a substrate and do the chemical reaction that it was supposed to do with its original substrate. And you can do that through protein, basically protein evolution or site-directed mutagenesis. You can either go through, okay, I'm going to evolve this protein through really directed evolution, which we've done with some. But the most promising approach, and the ones that show some interesting results, is actually designing the protein itself. So you go into AlphaFold and you check, okay, what if I change this? And that's how big will the pocket, the active site of the enzyme, how big will it be? What is the substitute I need to change it to have the types of interactions that I want, so that it doesn't take the first substrate, but it takes benzene. And this was actually quite successful.
You're going down all of these different experimental paths, simultaneously. Can you just give us some insight into, you know, what is the data that you're collecting as part of the design, build, test, learn loop to try to like stay informed about all of the paths that you're going down, and, you know, be really smart about the resources that you're deploying and the experiments that you're running to get yourself to the end goal that you're trying to get to, you know, as efficiently as you can?
So putting in place R&D processes is something that I love to do. And so we went through a lot of different ways to do parallel work, to iterate fast, trying to go at the speed of software with biotechnology, which is not, and plant biotechnology, which is not easy to do. The best system that we found that we currently use right now is working with projects that are defined in time and scope and objectives before we start the project. So we have right now the team something like 20 projects running at different stages of maturity. So you have early stage projects where we say, okay, we're going to spend 4 months to get to this first milestone with this time, this team, and this amount of like percentage of time of each of the individual contributor, these resources. And within the 4 months, well, after the 4 months, we have what we call we called it a great filter. So this is where we try to kill the project. So after 4 months, we already decided these are the KPIs. This is the advancement that we needed to have, the exact results that we needed. If we don't pass the results, we kill the project, which is something that we actually celebrate because it opens up resources to do another project.
Right.
And so you keep iterating this way, trying to make sure that the resources are as well distributed as possible. So you keep a flexibility, but you know exactly at which stage you take a decision, and you let things, since it's very much an empirical journey, you let things go their way and see what's the most promising way that always goes through these great filters. So it goes through great filter 1, then great filter 2, and then the project actually is done. So we, for example, a protein engineering project that you need to have a specific KM, KCAT, a specific activity if you want. And if you hit these numbers, you move forwards and you get to a higher stage of development. So you created this protein. Now it's about, can you make this protein work inside your plants? And then there's another filter afterwards. Okay, now can you create a stably genetically engineered plant that produces this protein that actually absorbs these pollutants faster? And then the last stage would be, well, is it fast enough to have a real impact in real life conditions? And so you have a lot of projects that die at the first great filter, which is what we want. And we really try to kill the projects. We incentivize people to kill projects, actually. So at the end, the only projects that go through and end up being ready for commercializations are the ones that survive, kind of a selection of the fittest in terms of projects.
Yeah, interesting. So, I mean, you gave the example of sort of a protein engineering problem. What can you give some examples of some of the other classes of projects and like the milestones that you had inside them to like understand how you were progressing across all of these different avenues?
So inside the Power Jobs, there's 2 strains of bacteria. One strain that's a Mycobacterium that can absorb formaldehyde. The other one is a Pseudomonas that can absorb BTEX, benzene, toluene, ethylbenzene, xylene. These are naturally occurring bacteria that like to live close to plants, either in the rhizosphere or in the phyllosphere, that forms a natural microbiome. So the entire microbiome project, we want, okay, first step is let's find organisms that are harmful for humans or animals and that can live in symbiosis with the plant and that can use BTX as a sole carbon source, meaning that it can grow with only these pollutants. So that was the first great filter. If you couldn't find these organisms, the project would die. And we found some of these organisms, one of them, Pseudomonas, that we use currently. The second stage was, okay, now, if you want to have an impact at scale, we have a estimation. So we run some calculations with data that we generate in-house. If you want it to have any sort of impact in real life, you need to have this efficiency, this remediation speed. And so there's another great filter 2, where, okay, now let's try different things, how to make it efficient. So let's try directed evolutions, so manually. So if you put the bacteria into media that are more and more concentrated in benzene, and the ones that survive all these different steps should be the ones that are the most efficient at using benzene as a carbon source. This is one path. The other path is also directed evolution, but another way. We take the same organism and you try to evolve it, not by putting it to more and more pollution levels and killing most of the bacteria, but you make it constantly grow on a low amount of benzene. So you select for the efficiency. So how fast can we pollute a small amount of benzene instead of how do you resist to high amounts of benzene? So this is another way. It's more efficiency of use of benzene. And then you have, let's use transgenesis. So let's insert transgenes from other organisms to add to this capacity. And then there's, maybe it's not about having the pathway being more efficient, but something else within the bacteria. So what if you could make sure that once the benzene is inside the bacteria, it never goes out. So it needs to metabolize it. So that's not directed evolution, that's specifically targeted mutation inside the genome of the bacteria to knock out the genes that code for the benzene to go out of the bacteria. So it has to deal with it, has to depollute it. So this is another great filter that we had to go through. And so which one of these projects are the best to move forward? And so we found the best way, moved forward, and we ended up with a strain that is orders of magnitude more efficient at depolluting these BTX. And so high enough efficiency to have a real impact in your bedroom, for example, if you put it on one single plant. So this is another example of different stages of projects that had to die for one of them to succeed. And then obviously it doesn't create a product. You need to add these other bacteria, Methyllobacterium, that went through the same exact process, put them together, make sure that they work well together, then make sure that these can form a stable microbiome, at least stable enough that doesn't die within 1 or 2 days. And then you have something that can become a product.
Obviously, throughout this entire process of experimentation, you're keeping your eye on the end goal of having a plant with a microbiome that can, that can metabolize these VOCs. What are some of the measurement methods or the North Star methods that metrics that you're using to understand whether you're moving in the direction of having the product that you're targeting, even as you're going down all of these avenues and sort of hitting these different filters?
Testing the efficiency of the product was the most difficult thing we had to unlock at Neoplants, because nobody has done that before. So all the VOC sensors that you have in your home, they don't work. They react to ethanol and not formaldehyde. So if you put perfume on, you'll see the VOC levels, the total VOC levels of your sensor shoot up. But if you actually put pure formaldehyde, like super high concentrations, it doesn't even react. So same thing with benzene. They don't detect benzene, they detect other things. Basically, the total VOC sensors right now can only tell you the air seems stuffy. This is what it says. Doesn't mean that there's harmful pollutants. It can be ethanol, which is not really harmful. It's not at the levels that it detects. So we needed to develop our own testing methods, and we needed to be high throughput and extremely sensitive, which is very, very difficult to do. So we don't have one testing methods, we have many. So talking about the microbiome, the first testing setup we've developed. So obviously there's the molecular testing, making sure that the pathway is working. This is kind of level 0 testing, but once you have the strain, you want to make sure that it actually absorbs these pollutants. So outside of analytical chemistry and enzymology, you have the bacteria, we put it inside the soil of a little small plant that you put inside a 2-liter jar. And we found sensors that are not very specific, but very sensitive to benzene, for example. And you put these sensors on the lids, like on the lid of the jar. So you can close the lid and insert specifically benzene inside the jar. So even if the sensor is not specific to benzene, since you only introduced benzene inside this jar, but everything it detects is benzene. And we have something like 20 of these jars in parallel. So you can test 20 different concentrations or strains or whatever you want in parallel with all the controls that you want. So this is what we call level 1 test, where you have these little plants inside 2-liter jars and see, okay, what works, what doesn't. Gives you a good sense, and you can compare them between each other. But the concentration levels are way too high. They're much higher than what you can find typically indoors. It's not the right setup. It's also a static chamber. It's completely sealed. It's only 2 liters. So you have then level 2 testing. This happens, so you take the best results from level 1, we go to level 2. Level 2 is a 35-liter chamber where you flow through air that is polluted. And this chamber, we have 2 of them running in parallel, control and experiment. They're linked to GCMS, so gas chromatography linked to mass spectrometry to be much more precise. But you can only do these measurements every 30 minutes or 1 hour. Otherwise, technicians go crazy. So it's a kind of passive, active sampling, not passive data collection. But we can do this, which we've done, but you can't do this at scale, but you're very precise. And you can actually now say, okay, this solution, if you flow through this amount of air, you have this efficiency. So you start to have a good idea and you can plug in some numbers in your simulation, because we can go into it. We've also created simulation on how can this work. And the last level of testing was 2 chambers that are 160 square foot that we've built in stainless steel. So there's no interaction with the walls. And in these chambers, we can control humidity, temperature, pressure, light source, amount of pollution. And we can track everything that's happening in these chambers in real time at parts per trillion sensitivity. So extremely sensitive, with a machine that's called PTRMS. I think it's proton transfer resonance mass spectrometry. So this machine alone is like 300K euro just to be able to. But it's extremely sensitive and works in real time. So we link this machine to these 2 rooms, and we've put inside these rooms IKEA furniture or any other furniture that emits or doesn't VOCs, or walls that we paint with certain paint. So you actually create an actual bedroom, same conditions, but you control everything, and you can test in normal plants or genetically modified plants, or plants with or without the product drugs. And you have extremely precise data with this. But one experiment takes a month to run there. So you can imagine that it's not high throughput. So we needed to go through this 3 level of tests, from 2 liter to 35 to a full bedroom size test, where every step along the way you lose throughput, but you gain preciseness.
Yeah, very interesting. And you mentioned the simulation process that you use to sort of understand the dynamics of what's happening in these tests. Can you just give us some insight into what that simulation looks like, and how the experiments that you're running sort of help you to understand the simulation and vice versa?
The simulation is very, very rough at first, and then you can add in more and more complexity as you go along. So at first, the first thing we've done is, okay, what is the typical airflow within a bedroom? What's the typical concentration within the bedroom? How much do we think the plants could absorb? And it was very rough. It was so it's something that we've done with, I would say, relatively simple equations. But the more you gather data, specifically, the more you set up your testing methodologies, and the more data you have on wood emission levels, how fast do they emit the woods, how fast your microbiome actually absorbs these VOCs, etc., you can actually create a more and more precise simulation. Now, it's something that we do. It's something that is going to be more and more powerful the more we go further along this process. We're really at the beginning of this process, because we have to kind of invent a lot of things or adapt a lot of different things from different areas, different fields, from the indoor air quality fields, the microbiology field, to plant genetic engineering. So you build the simulation, different parts of the simulation, little by little, and then you put them together. And you try to see, okay, does the simulation correspond to what you actually see? and it never does. So you have to go back to the drawing board and see, okay, what's wrong, etc. But at some point, you get to a point where it actually is useful to have the simulation. It's precise enough to give you a rough idea of some paths that you can say, okay, for sure, this is not going to work. Even if you can't predict exactly what is going to work, you can say, okay, high degree of confidence, it's not worth running these tests because this will not be useful.
Can you just give us an idea of what are like what's at what level is the simulation, or is it and what are some of the variables that go into it, and some of the trade-offs that you have to think about that the simulation might demonstrate for you?
Different bricks then come together. So this is how we think about it, because you can't have a whole simulation. So the first thing that we've done is air exchange within a room. We've done that with an external partner, and okay, you can have a simulation of the airflow that happens within a typical room. So this is one brick that doesn't require any data from our side. Another brick is once you get some data on the efficiency at which the bacteria absorb these pollutants, you simulate kind of a well that absorbed like an attraction force for VOCs. That's the plant. And you can plug in numbers for this like well of VOCs according to the data that you have with the 35-liter chamber or the VOC, like the actual full-size bedroom. And you try to refine them as much as you can. Another big part of the simulation that is currently not completely linked yet is the plant itself. So as we go along our genetic engineering of the plants, at first we assembled the genome, but we also had the transcriptome, the metabolome, a bit of the proteome. So we have a simulation of the metabolism of the plant. So we have kind of a virtual plant that has as much data as we can have with a plant that's only us study inside our computer, that allows you to say, okay, this enzyme is probably missing, so you probably need to bridge the gap with this additional enzyme. We're not at the quantitative level yet, but once we gather enough data, then we can plug in numbers and then plug in that simulation into the indoor air quality simulation. And there's something we're trying to do and to actually have to give to people is actually this simulation part in and of itself. So since we're generating a lot of data on how do furniture affect indoor air quality, and nobody is generating this data ever. Like we talk to the people at Dyson or at the EPA, like none of those people actually studied at this level of sensitivity the dynamics of the VOCs being emitted by different types of woods, plywoods, paints, etc. So we try to gather as much data as possible and then create an indoor air quality simulator that people can actually use to test or to predict the level of VOCs they have inside their bedrooms, since there's no good sensors available today. So these are different things that we have currently when we talk about simulations, and how do we try to predict the level of pollution in people's homes, and then how does our products affect this level of pollution.
Yeah, that's amazing. And I obviously we've gone through sort of the experimentation process, and then some of the assays that you're using, the ways that you're measuring the outcomes that you're getting, and then the simulation component as well. I'm wondering, from your experience, starting from, a very low data environment and sort of slowly building up the data that you need in order to characterize the system that you're creating or that you're trying to influence inside of people's rooms, inside of people's homes. What have you learned about sort of the limitations of data science or computational approaches in, helping you along the path of solving the problem you're trying to solve?
When I started this journey, I was very much like a wide-eyed, young, naive little scientist thinking that, oh, we could generate fantastic level of data and then build our models and then predict things, and then we'll have the computer tell us what to do. So it doesn't work like this at all. It's very much a step-by-step process, especially if you're the only people researching exactly what you're doing. So you can have a bit of resources here and there, but you need to generate these data. So at first, the first thing that I learned is you need to have a very, very precise test with very precise outcomes of these tests. And setting up this test and the right KPIs is the most difficult part by far. Setting up the test and making sure the test is the right one is the most difficult thing. And then running the test is actually the easy part. It's just about throughput and making sure we are creative enough to find a solution that actually works. So if I had an advice to give to my younger self is focus the first part of your R&D on just setting up the tests. Don't even try to create the product. Just set up the tests. And then once you know the tests are doing well, then you start creating the products. You'll go much faster there. So, and the results of these tests need to be in a format that's computer friendly, even when you start. So one example of the test. So since nobody has really tried to do genetic engineering of that plant before, we need to understand what can drive once you find the genes you want, what can drive their expression. So there was a huge amount of work on which promoters work on that plant, which terminators work, and the combination of the two. And it didn't express high enough. So how can you increase the expression level? What if you add two terminators? And then, okay, once you explore all these possibilities and create even synthetic promoters and all these things, how do you test the activity? What's the actual test? Do you need to wait for the entire stable plants to grow to be able to test the expression level? And that will take a year. And you can't test it on another plant because it has completely different metabolism and genetics. So what tests can you run that will last a week or two and give you good enough results with high enough throughput that can predict, okay, once the plant is stably transformed with this promoters and terminators and this gene, it's this level of expression. So we need to say, okay, what's the fluorophore? What's the protocol to do what we call agroinfiltration? So to transform in a transient way the genetics inside the leaf. And how can you normalize these things? Like there's you need to control for a lot of things, because if you transform more cells and you just look at the fluorescence level, you have a high fluorescence level, but each cell has very, very low expression. So if you have one cell that expresses a lot, then your problem is not the expression level, it's the fact that you can't transform the other cells around it. So even like that, like, okay, you have your fluorescence outcome, but even then you need to say, no, I also need to count the number of cells that produce this fluorescence that will give you an idea of the expression of the strength of the promoter and terminators. And then you need to say, okay, now I'm going to replace my fluorophore with my gene of interest. Am I sure that the promoters and terminators are actually working when I replace the gene? Does the gene have any effect on the activity of these regulators? Literature was very ambivalent about it. We found out that yes. So what kind of test can you run if there's a difference with them? If you change the actual enzyme, your reporter fluorophore doesn't work as well. So this is the type of challenges when biology needs data science, is how can we have the best test possible that's reproducible enough to give you high quality data enough to really inform what's the next step in your R&D journey. And the only way to craft it is actually to go through it and see, okay, this doesn't work, what does work, etc. And at the end, you get to, once you generate a lot of data, you can extract a few learnings from it, a few general rules, a few general principles, and so be more refined in your approach. But you can only deduce these kind of rules of engagements of the genetics of the plants if you have the standardized test, and if you have in high quality enough data to, even if you don't think about simulation level, just enough data to lead you in the right direction or to remove some path that we know will not work. So it was really a step-by-step process.
Yeah, that makes a lot of sense, and I think that what you're saying gets lost in a lot of the conversation about sort of foundation models for biology as compared to foundation models for language, which is that, obviously if you have data of a certain scale or there's zero marginal cost to produce new data, then noisy data can teach you a lot about the world. But when you're in an environment where there is marginal cost to creating new data points in both time and money, the design of that particular measurement system and ensuring that you're measuring the thing you need to measure and on the time frame that you need to measure it, in order to lead you in the direction of what the next experiment looks like, that becomes so much more critical. So I really enjoyed hearing you describe that.
I think the most difficult challenge that AI will face is putting the results into its right context. Okay, let's say you want to have the AI that can predict protein activity. Okay, we can predict protein activity with AlphaFold, you can do some predictions, but that's in a vacuum, that's in vitro. When you talk about a full organism, what's the effect of pH and all the other things on the enzymes? What's the effect of the expression level? How does this enzyme react to even how does the gene react to the transcription machinery of the cell to produce this enzyme? Is this enzyme stable or not? In this specific context, where do you want to put the enzyme to be active in the cytosol, in the mitochondria, in the chloroplast, in the nucleus? I don't know. And then is every single cell, so you modeled the effect of your enzyme in one cell. Does every single cell express this enzyme? And even if it does it react with other stuff from other cells? What are the different organs of the plants and how would they react with this stable transformation? And how can you predict this? And then, okay, now that you predicted the expression level, even the phenotype of the plant, so what's the effects on the actual function you want to add to it? So it's not a plant in a vacuum, it's a plant at this temperature with this amount of sunlight that has this effect on its physiology, that is watered this amount of time in people's homes. So if we want to talk about the dream of having an AI that can predict, oh, you want to have a plant that purifies the air from formaldehyde, this is the pathways, these are the enzymes. You need to integrate all this different context to predict, yeah, this is the way to go. And this is just a context that we know about as scientists, which is, I don't know, 1% of how the organism actually works. So at first, you need to understand biology, then you need to model it, and then you can predict things from it. And understanding is basic research, and we're very far from really completely understanding it. So I believe that machine learning, just AI models in general, will be very, very efficient and are already very efficient at very specific tasks that are very far from solving biology and predicting what type of changes you can introduce inside organisms to have this type of impact. This is still a very empirical-led research. But I do believe that we'll get there. I do believe that AI is going to change everything when it comes to biology, but the challenges are insanely high.
I'm interested in how you think about avoiding becoming the sort of insular R&D focused organization, about balancing the engineering performance of and the biological stability, the research oriented outcomes of what you're trying to accomplish with the product focused outcomes, the manufacturability, the scalability of what you're trying to do.
It's about creating a certain culture within the company, and it's little things. For example, we have a monthly company review where we talk to the entire team, business and R&D, about what's happening, updating everyone about the different things. But it can go even like bottom up. So for example, we involved, I think, almost all our scientists in the customer support the side of things. So they can actually talk to the customer that is using the product. And so it's super motivating for the R&D team to know that, oh, our product is out there, people are asking about it, etc. And I can help them use the product. It's not something that we're doing at scale, but it's something that we intentionally have done, at least at first, to bridge this gap between the bench to the bedroom. So it's something that we're very, I think, successful with. Everybody in the team thinks in a product kind of way. What's going to be the impact? What even most people in R&D don't even think about the products and even less about the customer experience. So we try to do both. So, okay, it's really cool. You found your fantastic little organism that does what you want. But can this organism live with indoor plants? If not, what are we even talking about? So all these, you need to have this culture within the R&D team of always think about the end product, and even more, the user experience. And so this is how you bridge the gap. You kind of loop the loop back. So once you have first product on the market, you want to have feedback, and you want the feedback to go back directly to the R&D team. So you actually get the R&D team to ask for feedback directly. We do this at scale in a more data-oriented way, obviously, with our marketing team. But this direct link, I think, is important. So I think it's these types of little things that you can inject into the culture of your company, the more it faces the market. And it's very useful because then R&D people can say, oh, I've heard that because consumers want to do that, and actually you can do it. And sometimes the marketing team or the kind of the voice of the customers hears the customer saying, oh, I'd like the I would love for the power drops to smell good. And the marketing team, yeah, but this is not what we do. We do indoor air quality, air depollution. We're not interested in fragrances. But if the R&D guy hears it and says, huh, that's easy. I can just introduce one gene that produces a smell. Why not do that? So we can then have a discussion between R&D and marketing saying, well, actually, this is actually not very difficult to do in the next iteration of the product. So I think R&D needs to be in constant contact with the market. And this is how we build this product creation machine and not just a R&D institution.
Amazing. Well, what do you say to people who have some discomfort with the idea that you're genetically modifying these plants, even if they're in support of the reason that you're genetically modifying them?
There's a lot of jokes that I could do. Like if they have dogs, do they know that their dogs are genetically modified wolves? But in a more serious way, I think the most important thing is to try to educate the public about what is genetic modifications. And it's very, for me, it's very simple. Is we've been genetically modifying organisms since civilization existed. The first technology, genetic modification technology that we've developed is called domestication. We domesticated crops and dogs and sheep and cows and a lot of different organisms that are not supposed to be the way they are. Cows are not supposed to be these big lumbering sack of meats that are super docile that you could just come over and just slit the throats and eat. The maize, corn didn't used to look like this. Tomatoes are monsters compared to their original form. So it took us 10,000 years to create these organisms, but they are definitely heavily genetically modified. But people don't think about it as genetic modification because it's not artificial. But it's 100 years ago, we've introduced artificial genetic modification through what we call mutagenesis. And it's random mutagenesis. You put UV on your tomato, let's say, because you want to create a tomato that's a bit more red. So you put UV into your tomato seeds, you generate a lot of, like millions of different mutations, and then you select the seeds that produce the plants, the tomato that's the most red. You have thousands, millions of mutations inside your genome, but it's not considered GMO by any country in the world. And you have heavy genetic modification. Why? Because if you outlaw this, you outlaw agriculture. Every single seeds in the world is produced using this method of random mutation and then selection of what you want. So this is kind of the second level 100 years ago. Now, 50 years ago, or 40 years ago, we've developed a method called transgenesis, which is, instead of doing these random mutations, you can insert one single gene inside of it. Now, the big question is, what is the function of that gene? And this is where you realize, is a GMO good or bad? GMO is like a technology. It's like, is metallurgy good or bad? Well, it depends. If you're producing swords to kill your neighbors, it's bad. If you're producing casseroles to feed people, that's good. So GMO is not good or bad in and of itself. But the first applications of GMOs were to put herbicide resistance into maize and put so much herbicides in the fields so that the only plant that could survive are your genetically modified plants that are full now of herbicides and pesticides, because you put it all over your field, destroying the ecosystem, the soil health, and it's bad for your health because you're eating herbicides and pesticides. But the fact that it's GMO in and of itself is not the problem. It's the application of these herbicides and pesticides. It's too bad that the first applications are those that are in service to these big agricultural companies to sell more herbicides and pesticides. And this is their revenue model. So this is called transgenesis. But if you insert a gene that is not a resistance gene to these herbicides, but a gene that purifies the air, why would it be bad? These are all natural things in the sense that it's a naturally occurring gene that I introduced from one organism to another. You can call it Frankenstein, but it's when you create a light bulb, it's not supposed to be there. You're like the only natural source of light should be the sun and the stars. Why are you creating this other source of light? At some point, we need to be okay with, well, we have two paths in front of us. Either we work with nature or we're destroying it. There's no other way. And the way we've been doing it currently right now is destroying nature instead of leveraging its power to try to find sustainable solution. And nature is sustainable by nature. So why not use it? And then the last application, the last type of genetic modification is CRISPR-Cas9, genome editing. This is cutting bits and pieces of the genome, putting them back together, changing little things inside of this. And people are much friendlier to that approach because you're not introducing something exogenous to the organism. It's even that currently the European regulation says that this is a GMO. Even if you introduce one single deletion, that's a GMO. And whereas the millions of mutations you've done through UV, it's not GMO. I think to answer, like it's a long-winded answer, to answer your question, what would I say to people that are iffy about GMOs is learn about genetics. It's beautiful. It's elegant. It has so many applications that you don't even think about. All the insulin that people have right now are produced by a GMO bacteria. That's the only way we have to produce insulin, is to introduce the insulin gene inside a bacteria and produce insulin. That's all. That saves dozens of millions of people's lives every year. So it's a type of technology with bad and good application. It can either save the world or possibly destroy it. So we need to have the right discussion. It's not about is GMO good or bad. It's about what application do we want to promote and what application do we want to ban.
Yeah, not what is the tool, but how is the tool being used? Well, you've been very generous with your time today, Patrick. I really appreciate it. So before we let you go, where can people go to find out more about your work or connect with you online?
Yeah, so you can go on neoplants.com. This is where we sell our products. If you want to support what we do, go there, try it out for yourself. And I hope you'll feel much healthier, especially if you have asthma or allergies. This could be very beneficial and will help us continue to pour these resources into the deep tech R&D projects. So just go on neoplants.com. You can check out also the science we have. We written a white paper that explains everything about the problem, the technology that we use, the solution, the raw data. We try to be as transparent as we can about the science behind what we do and how do we test it. So yeah.
You've got one happy customer over here. I've got my Neoplant sitting here right next to me. We renovated our house and had to paint the interior. And so getting VOCs out of the air is very close to my heart, and love the work that you're doing at Neoplants. So thank you so much for joining us today, Patrick. Really appreciate it and look forward to connecting down the line.
Thank you very much for inviting me. It was a very fascinating discussion.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.